State University and one of the coordinators for the webinar series, along with Robin Usborne with Michigan State University and Jody Ellis with Purdue University. Today's presentation deals with thousand canker disease on black walnut and is presented today by Dr. Ned Tisserat, plant pathology professor and extension specialist. BS from Colorado State University, his MS from Texas A&M University, and a PhD from the University of Wisconsin-Madison in plant pathology. He is an extension specialist and professor in the Department of Bioagricultural Sciences and Pest Management at Colorado State University. His research includes studies on the biology and management of turf grass, trees, ornamentals, and small grain diseases. His research group is currently studying the biology of thousand canker disease of black walnut. Continuing education credits will be available for this session. If you need verification that you attended this webinar for other purposes or for self-reporting, please indicate that by sending an email to the address shown at the end of the presentation. Feel free to type your questions and comments into the chat feature on the webinar screen. Your questions will be written down to keep the presentation moving. Ned will answer those questions after his presentation. This webinar is being recorded and will be available for viewing on the emeraldashboard.info website, all one word. This presentation and all other past presentations are up there. Thank you for your interest today, and please feel free to email any questions or thoughts you have about today's presentation. We always like to hear if there are suggestions that are helpful or any other recommendations that you may have. With that, I welcome Ned, and feel free to start your presentation.
Okay, am I on?
Yes, I can hear you.
Can you hear me? Okay, let's get started then. Thank you very much, and what I'd like to do today is discuss thousand cankers disease of black walnut, and what I'm going to present today is a compilation of our experiences here in Colorado and in the West, and this is information that both Dr. Whitney Cranshaw, the entomologist, and I have put together. This presentation has been uploaded on the website thousandcankersdisease.info, so if you want to use portions of this presentation in any of your meetings, feel free to do so. I just want to start out so that everyone's on the same page. I'm going to be talking about thousand cankers disease of black walnut, and this is a lethal problem that we have observed here in the western United States now for, well, upwards of 20 years, it turns out. For most of the time we didn't really know what was going on, but it's only been within the last 3 or 4 years that we've kind of figured out what was happening to our trees. I want to stress that this is a lethal disease, at least in most of the western United States, and I have no reason to doubt that it's going to behave just the same in Tennessee, and that's an unfortunate fact. I hope I'm wrong, but I doubt that I am. So what I'd like to do this morning is just give you a brief history of thousand cankers disease in the West, and I want to spend most of the time on diagnostics, and I'll spend some time talking about how you would go about a windshield survey, how to look for symptoms of TCD from let's say a car or walking down a street. I'll spend some time identifying on how to identify the walnut twig beetle and the fungus associated with the beetle, a little bit of time on how to collect samples, and then, as you probably know, there are things that could be easily confused with thousand cankers disease, so we'll go down those topics. I know I'm speaking to the choir here, but just as a little background information, black walnut is probably one of the premier hardwoods in North America, and its native distribution runs from the Atlantic coast into Kansas, Oklahoma, and Texas. But as the pioneers moved west, they brought nuts with them and established black walnut in many small towns throughout the western United States. And it's very difficult to go into a small town, let's say in eastern Colorado or in Utah, and not find black walnuts planted. And I suspect as you go through your municipalities looking for 1,000 Cankers, you're going to be surprised at how many black walnuts are present in towns. I got involved in this problem in about 2006 here in Colorado, but the problem actually spans back to the early 2000s, and the foresters in both Colorado Springs and Boulder began to see decline and then eventually mortality in their black walnut trees. And the second woman from the right is the forester in Boulder, and she is really the one that alerted us to the problem. And so she really needs to take, well, she gets all the credit for initially finding this disease. Since the early observations of this, the disease has continued to spread in Colorado, and we now have mortality of thousands of trees in the Denver-Boulder area. Just to kind of give you an example of how the epidemic is progressing here in Colorado, and to relate it to a historic disease, I put up the progression of Dutch elm disease in a municipality, and I don't remember where I got this. It was from some town in the Midwest. And as you can see with Dutch elm disease, the incidence starts out fairly slow, but like most epidemics, it increases exponentially. So after 6 or 7 years, 40% of the trees in this particular municipality were killed. And if you look at what's happening in Boulder, it's got the same kind of pattern to it. We started out with noticing some dieback in 2001, and by 2010, 60% of the trees have been killed. One interesting point here is that the trees that have died have been the larger diameter trees. Trees that are remaining are, for the most part, less than 10 inches in diameter. So if you're looking for this disease, I would suggest that you look in the larger diameter trees first, because contrary to the name of the beetle, this beetle appears to prefer attacking large diameter trees. I want to also stress that this disease has not been limited to Colorado in the West. We actually believe now, based on historic records, that 1,000 Cankers disease originated along in the Wasatch Valley of Utah and in the Willamette Valley of Oregon. And there are reports of walnut decline and dieback in those two locations as far back as the early 1980s. I also want to stress that this is a progressive disease and it's continuing to spread in the West. We just recently had reports in Walla Walla, Washington to the north, and Albuquerque, New Mexico to the south. So this is important to us because initially we thought that these outbreaks were related to drought, but the scope of this problem in the western United States probably makes that assumption a false one, and I'll talk later about that in a moment. I put this map up to give you an idea of what the distribution of 1,000 Cankers disease is in the western United States. The dark blue counties shown here are ones where we have found the walnut twig beetle and the fungus on eastern black walnut and have confirmed 1,000 Cankers. The light blue counties are counties where black walnuts have been reported killed, but the trees were removed before we could sample them. So some of those counties, let's say in Utah, where the trees were killed back in the early 90s. We have also surveyed a juglans major or Arizona walnut down in New Mexico and Arizona. Those are represented by the red shaded counties. And we have found the walnut twig beetle and the fungus in all locations that we've looked. But on Arizona walnut, it's not causing a major problem. So we believe that Juglans major is probably the source or the origin of the beetle and the fungus. The yellow counties in New Mexico represent areas where we've surveyed little walnut or Juglans microcarpa, and we have not found the beetle or the fungus there. And then there's some dark, sort of pinkish counties in Texas where I surveyed for the beetle in Arizona walnut in these isolated pockets, and I could not find the beetle or fungus in those areas as well. And we have surveyed throughout Colorado, and the delimitation of the disease is pretty much as shown by those dark blue counties. I'm not going to go into any detail on this, but there are also two other walnut species in the West. One is California, Southern California black walnut, and Northern California black walnut, and we have found Dr. Seibold in California has found the beetle and the fungus associated with decline of these trees as well. That's a whole different and very interesting story, but I won't go into that this morning. And the reason we're all listening to this today is not what's happening in the West, as tragic as it is. It's the recent confirmation of thousand cankers disease in Tennessee. I was able to go to Tennessee and look at these trees, and everything that I saw illustrated or demonstrated to me that the progression of the disease was very similar to what we see in the West. Based on the distribution of the disease in Tennessee, we suspect that it's been there for many years, possibly 5 or more years, and it's over a fairly large geographic area. So this is not something that we're going to eradicate, in my opinion. So we have to try to mitigate the movement of infested material to other parts of the country. Okay, that's enough with the historic aspects of it. What I'd like to do is just kind of give you an overview of some of the symptoms that you might look for as you're driving or cruising looking for damaged trees. And the problem with TCD is that the symptoms can be easily confused with other problems. And in addition, some of the initial infection occurs up in the crown, and sampling is going to be a problem. One of the earliest symptoms that we observe in infested trees is a flagging of individual branches, and I think you can see it with this walnut in the upper right hand corner. There's just one individual branch where the leaves are turning a bright yellow. This type of symptom commonly occurs in late June, July into August. You've got to be careful if you're looking for these kinds of symptoms past mid-August, because you can get natural senescence that will look very similar to this. This flagging may be scattered in the tree, but more commonly we see it on a single scaffold branch, because the beetle tends to attack the tree non-uniformly. But remember that this can be confused with many other problems. We see squirrel damage here in the western United States that would look identical to this. So while this is helpful, it's not diagnostic. Here's just another example of showing you the kind of damage you might look for. Notice that in this tree, most of the branches and foliage look very healthy, but there's one individual branch where you get a little bit of flagging, as illustrated by the top arrow, and a little bit farther down. I'll talk about this in a moment, but if you were to cut that branch off and look at the twigs, you would not find anything. So it's very important how you sample these trees, and unfortunately I think you're going to receive a lot of samples that are going to be inconclusive because people aren't going to sample them properly. Another symptom that we associate with TCD is the thinning of the upper crown. In my opinion, a healthy walnut, you should not be able to see sky through the canopy. So if you're looking at a tree and you can see sky, that's an indication that something is going wrong. Now, it could be a root problem. There are many, many other things, but in this particular tree, you can also see that there are several flagging branches. So this would be about as early a stage as you would be able to detect 1,000 cankers. The question then becomes, how long has this tree been infested? And we still haven't determined that to my satisfaction, but I would say this tree probably has been infested for at least 1 or 2 years. This type of symptom where you get collapse and wilting is not as common in my opinion, but you will see it, and in cases like this the foliage will dry up and remain attached to the tree. I find more commonly that it's instead of seeing this type of symptom in midsummer, this branch would just be thin but then fail to leaf out the following spring. So while we see this kind of damage in the early stages, it's not nearly as common. It's just individual flagging of branches. Okay? Another kind of symptom that we see, the leaves may flush out and then collapse. Notice the picture on the left, the arborist has tried to prune out the dead areas to no avail, and the tree is very thin. And in the picture on the right, you may get flagging and death after leaf emergence. I'm just going to show you a few images of stages of decline and not comment too much on these. The picture on the right is very characteristic of mid-stage decline of a walnut. That tree would probably be dead by the following spring. Here are some other examples. One important point here is that the bark does not slough off the tree. And if you're looking for, when we talk about cankers, if you're looking for open-faced cankers, you will not see it. The cankers are all under the outer bark. We do see resprouting of these declining trees. The sprouts will also die back. Apparently this is not as common in Tennessee or in Oregon, but it is something that you can look for in your surveys. Just to give you an example how quickly this occurs, the tree on the left, the picture was taken in June of 2008, and by the following spring that tree was dead. What we don't know is the time between initial infestation and development of symptoms, as shown, let's say, in the tree on the left. And that there's argument, vigorous debate about this. I say it may be 3 to 4 years. Others say it may be upwards of 5 to 10 years before you see any symptoms in a tree. So that's something we have to clarify. But it certainly makes your job more difficult in identifying the problem. Okay, that's kind of what you would look for in a windshield survey. If you're looking for the beetle, good luck. This is a very tiny beetle. It makes the emerald ash borer look like a monster. It's about 2 to 3 millimeters long and about the size, diameter of a pencil lead. We do know, or we suspect, that this beetle is native to Arizona walnut, as I mentioned. And in Arizona walnut, it usually goes in a leaf scar and creates this star-shaped pattern with a little bit of discoloration, but that's about as serious as it gets. It can help kill senescing branches, but for the most part it's not associated with killing trees. The walnut twig beetle has a couple of identifying features. As I said, I'm not an entomologist, so some of these are very difficult for me to see, but what I suggest you look for are these concentric rows of what are called asperities, or hair-like structures on the head capsule. The other thing to look for in the female is this beard-like or mustache-like structure at the front of the head. Those are the characteristic features of the walnut twig beetle. I'll show you slides in a moment. You've got to be a little bit careful here because there are some other things that you can confuse with the walnut twig beetle. But the main thing is look for these concentric rows of asperities. The holes of these are very small, as shown here. If you're looking for walnut twig beetle entrance or exit holes, look on the bottom side of the branch, not the top side. And you may get these little cracks associated, as shown in the bottom right-hand picture. But also good luck finding these holes in the main trunk. Now, I want to clarify something here. This beetle is called the walnut twig beetle, but on black walnut it goes right into the main trunk, and you can very rarely find it in the small twigs. So don't spend your time looking at small twigs. You want to look at branches that are 1 inch or more in diameter. And I would also suggest, if you're over 55, bring someone much younger with good eyesight, because I cannot, at my age now, I cannot find these beetle galleries. They are that small, and they are wedged into the crevices of the trunk. You may see a little bit of discoloration around the holes, as shown in the picture in the center. That can be helpful in diagnostics. The picture on the right is an English walnut, where you can clearly see the beetle galleries, or the beetle holes with the discoloration. Now, English walnut isn't super susceptible to this disease, but in our neck of the woods, the pressure is just so high that it is also killed. But you can see where the beetles have hit that main trunk. If you peel back the outer bark, you will see the meandering galleries in the phloem. This is not like emerald ash borer, where the galleries are right in that cambium phloem interface. So you've got to be careful here, because you can shave right through or right over or under the galleries. Notice the meandering galleries, and in the picture in the bottom right, you'll see a little white substance. That is the Geosmithia fungus. Also note the discoloration and maceration of the bark that occurs around the galleries. That's very characteristic of this disease. Where do you look for the beetles? Well, the beetles are much more common on branches greater than 1 inch in diameter. Don't waste, as I mentioned before, don't waste your time looking at small twigs. And I know you're going to get samples from clients wanting to know if something is walnut twig beetle. But unless you get branches that are more than 1 inch in diameter, your chances of finding the beetle are very slim. Also, you have to scrape off the outer bark to really see the galleries and to see the damage from the fungus. In this picture, all that brownish area is the discolored area is caused by the fungus growing out from the beetle galleries. The cream-colored tissue is the healthy phloem. So you can see the amount of phloem that's been killed as a result of the beetle galleries plus the fungus. And it's that combination that's really damaging the tree. Okay. Another problem. The beetles aren't uniformly distributed in the tree. You're going to find the beetles more common on the lower side of the branch, and it may be more common on one side of the trunk. And in my experience, I tend to see more on the sunny side of the trunk. Maybe here in Colorado that's important in the fall for breeding purposes. The picture on the right, what I've done is I've taken a log and I've just peeled off the outer bark, but I've left the inner bark or the phloem. That's not wood you're seeing. That is the phloem tissue. So you can see the individual cankers on the east side of the trunk and how the fungus has spread out. But what happens is that as more galleries are formed, those cankers coalesce to kill larger and larger portions of the bark. And that's the problem with this, is that you get that combined activity. The picture in the bottom left is also showing how these cankers coalesce to kill patches of the bark. And that's what you would be looking for when you're sampling. But again, you've got to get branches that are at least 1 inch in diameter to really see this kind of pattern. Here's a close-up of sort of a mushed up adult Pityopterus and some larvae. The larvae look like any other beetle larvae to me, but I'm not an entomologist. But notice the maceration of the bark and the wet appearance. That is very common in the advanced stages of this disease. You'll get this kind of soupy, macerated bark. And the beetles apparently aren't inhibited by that, because we can find numerous larvae, hundreds of larvae, developing in this kind of macerated bark. I don't have time to go into it, but it actually appears that the larvae are attracted by the fungus. And the fungus is somehow providing protection or nutrients or something for the insect. If you have a hand lens and you peel off the bark, you can often see this kind of dusty whitish appearance in the beetle galleries. That is the Geosmithia fungus that's fruiting right in the gallery itself. The picture on the right isn't a very good picture, but that is a pupae of the walnut twig beetle in a pupal chamber, and that whitish coating on the outside is the Geosmithia fungus. It's not a parasite, so that fungus is somehow protecting, or that's my assumption, is protecting that pupae, because that pupae would go ahead and mature into an adult. Look at the amount of the galleries in the phloem of these samples. Now the picture on the left would be a tree where maybe it has 50 to 60% crown dieback, and this particular sample came from the trunk itself, not from a branch. In the final stages of the disease, the phloem just becomes honeycombed with galleries of the walnut twig beetle, and the maceration of that tissue is complete. So if you see something that looks like one of these two pictures in the phloem, that would be a good indication that you have the walnut twig beetle. We have seen in some samples from Iowa and from Minnesota that there are galleries in the outer bark, or the dead portion of the bark, and those galleries look somewhat similar to these, but they are caused by some other insect, and to be honest, I don't know what it is. But so if you're looking just at the dead outer bark that's sloughing off anyway, you got to be a little bit careful, because you can see galleries that might look very similar to this. Okay. I can't overstate the number of beetles associated with these trees. These are two logs that you could easily fit into your fireplace, and we put these into a rearing chamber. And my colleague, Dr. Cranshaw, had his students collect all the beetles that emerged from these two logs, and that's what's represented in the vial. You can see this sort of dirty brown liquid at the bottom of the vial. Those are actually the corpses of the beetles, and Dr. Cranshaw forced his students to count all these. I'm glad he made them do it, not me, but he got 23,000 beetles out of those two logs. So you can see the potential for moving any type of wood with bark attached as a source for beetles and the fungus. It's just outrageous. It's just phenomenal. You know, one of our hopes was that since this is a beetle that's native to Arizona and New Mexico, it might not be winter hardy. And we've been observing this for the last several years. And in the Front Range of Colorado, we don't have sustained low temperatures, but we can get pretty darn cold. And this past winter, we got down to -20, and we found larvae, pupae, and adults in the trunk of infested trees. So don't pin your hopes on cold temperatures killing this beetle out, at least not in most of the range of black walnut. Now, you got to be a little bit careful here because there are other insects that can look like walnut twig beetle. Here's an example. The walnut twig beetle is on the left-hand side. Notice the asperities on the head. The beetle on the right is an introduced ambrosia beetle that we find very commonly now in declining trees. And this Xylobarinus has many similar features. There are some characteristics that would separate them out. I won't go into detail because I'm not an entomologist here, but that would certainly be something that you should make any kind of surveyors aware of, is that these beetles can look the same. Now, one difference is that the Xylobarinus, the ambrosia beetle, the galleries go into the wood. So if you see something like this, where you see the galleries going deep into the wood, that's probably a Xylobarinus. And I have a question here. What are the asperities? And the asperities, I don't know if I have a pointer, but if you look at the head capsule of this walnut twig beetle on the left, those concentric rings of little hair-like projections are what are called the asperities. You know, this is what I'm talking about right here. And right at the top of this female, you can also see this brush-like structure. That's also characteristic. Whereas the ambrosia beetle doesn't have that kind of structure. Okay? And that's the gallery of the Xylobarinus. As far as we know, the Xylobarinus doesn't transmit the geosmithia fungus. We haven't been able to isolate it. But it does transfer another fungus called Fusarium that can be associated with the decline. Here's the problem again. This is a tree that I saw in Tennessee. Looks perfectly healthy. I walked up to one of the branches on the underside, noticed the little depression here. That's where the twig beetle first hit, and the small canker surrounding that gallery. And the question here, I guess I was supposed to save the questions for the end, but we believe that the beetle is indigenous to New Mexico and Arizona on Arizona walnut. That's the best, based on genetic studies from Dr. Steve Seibold. That's what we believe. Okay, in looking at these trees, you know, if you want to sample for beetles, at this stage you may or may not find the beetles in the main trunk. That's a problem, because otherwise you have to get up into these branches on the right hand side and sample for them. So even up to 25-30% crown damage, you're going to have to get up in the tree to get samples to confirm. That's going to be a real problem in sampling for this. At this stage, you should be able to easily find the beetles in the lower trunk. And what you can do here is just take a hatchet and cut out a window, and you should be able to see the galleries very easily in a situation like this. Oh, the trapping is a mess. I personally would suggest that you not even worry about trapping. Using yellow sticky cards is an abysmal failure, much like trapping for emerald ash borer. Here's an example. Most of the beetles that we catch are in late summer, but we put these traps out in a yard where there was a stack of walnuts, and if you look at the numbers, we could only catch 40 beetles in a whole month in a yard full of diseased trees or infested trees. So that's very poor. So your chances of collecting beetles on trees out in the woods is virtually zero, in my opinion. Dr. Steve Seibold in California is developing an attractant, and apparently he has a very good attractant, and it's one that might be available in a year or so. So I think we just have to wait for that. Okay, so how does this beetle kills such a big tree, and as I showed you in previous slides, there is discoloration around the galleries. This discoloration doesn't look so bad until you start getting a lot of it, and then you get coalescing cankers, and eventually you get large areas of the bark dead. The fungus is very easy to isolate from any discolored phloem. Boy, I was going to show you a picture of the fungus, and it didn't show up on this slide. As a pathologist, I'm obligated to show you what a fungus looks like in culture, but for most of you, you'll never see it anyway, so I guess it doesn't really matter. A couple of things about the fungus. Don't shave too deeply to find this discoloration, because it's in the phloem, not the cambium or sapwood, at least not initially. And this is one of my favorite pictures. The picture on the right shows a cross-section of walnut, and walnut has a very thick bark. And I liken it to a multi-level apartment complex for beetles. So you can have beetles at various depths in the phloem, and as a result you have cankers forming at various depths. So it's not a uniform situation. Instead you get more what I call a marbled appearance to the bark. Eventually, however, most of that phloem tissue is killed, so you just have these little islands of surviving phloem. And that process is what causes the decline in the tree. Okay. Eventually this fungus gets down to the cambium. It will kill the cambium, and then you get cankers that look just like any other canker pathogen, you know, butternut canker or whatever. The difference here is that those cankers that I'm showing you there are under about a 1/4 inch of bark, and the only way you're going to see this is to shave off all of the bark to look for these cankers. In the bottom hand picture, notice that whitish appearance. That's the Geosmithia fungus sporulating. But I don't think that's going to make any difference, because that's, again, covered by bark, so those spores aren't going anywhere. The only way this fungus is getting around is by the beetle. An important point is that this beetle and fungus are killing the phloem, not damaging the wood. So if people are frightened and they want to preemptively harvest their trees to get around this problem, recommend that they don't do that, because they can still salvage their trees even if there's damage. And again, the important point here is the picture on the right shows a canker where the bark is sloughed off on an ash tree. That's Nectaria canker. But you're not going to see that with walnut. You're going to have to use a draw knife or something to shave that bark off. Okay. All right, I need to kind of speed up because I want to leave some time for questions. You're going to have lots of equipment for surveying for this. This sort of summarizes the death of the tree, and I'm not going to go through this, but basically it's a death spiral where you get more and more beetle hits, more and more canker formation, and then you get secondary things coming in that can finish off the tree. So my tips for field diagnosis is look for patterns. If you look up and see a lot of sky through the crown, something is wrong. And look for scattered dead branches, especially where dried leaves are still attached. I suggest just looking for the beetle, because if you find the beetle, I've never seen a situation where the beetle is separate from the fungus, so you just assume that the fungus is there, and it's easier to find the beetle and the galleries than it is the fungus. So that's what you should be hunting for. We think the best time to look for this disease is after the trees have fully leafed out, up until August, and after that time the trees start to senesce. It's amazing how many times people misidentify trees. In our area, Tree of Heaven is the #1 misidentified black walnut tree. So we do have problems with that. We don't have any hickories out here to speak of, so I don't know if that's going to be a problem. And if you get too late in the season, it can be tricky because of senescence. I'm just going to show you a few other things that might look like TCD. We've here in Colorado, we've had some problems with early fall freezes that damage walnut, and it will cause dieback. Another thing that keeps being brought up is drought. That's what we initially thought in Boulder, and Tennessee went through a bad drought, and that's what declining trees were diagnosed as. So if you blame something on drought, be careful. I don't suppose you're going to be blaming anything on drought this year, but another canker problem that we see on black walnut is fusarium canker. These are very elongate cankers, open-face cankers, and I don't think you're going to confuse this with thousand cankers. But black walnut isn't very susceptible to butternut canker, so make sure you identify the tree properly. One of the problems that could be a problem in your neck of the woods is anthracnose, because you can get severe defoliation in some years. So think about looking for foliar diseases as well. Collar rots, root rots are not going to be common in well-established older trees, but it may occur in plantations. And there are all sorts of other things, you know, storm damage, insect damage, herbicide injury, you name it. So it's not going to be an easy task because the symptoms that we initially see aren't diagnostic. I stress this because it makes so much a difference. If you're asking people to collect branches, make sure that they collect something an inch or more in diameter. I would get something 6 inches long, try to sample live but symptomatic branches. Don't remove the bark for shipment because the bark discolors rapidly on exposure to air. We want people to wrap samples in 2 plastic bags, and if you're going to mail this to a diagnostic lab, what I suggest is throw the sample in the freezer overnight to kill the beetles prior to mailing. That doesn't kill the fungus, so we can still do the isolation. It's just a way to make sure that you're not going to let the adults escape from one location to another. I get this comment quite a bit. You know, people say, well, it's a problem in the West, but it won't really be a problem in the East. And then it showed up in Tennessee, and people said, well, it's in Knoxville, but it probably won't get into our woods. And I'm hoping people are correct, but everything I've seen goes against this hypothesis. I end here with a picture of this tree in Eugene, Oregon. Look at the size of that black walnut. It's a monster. And I'm thinking, is that a tree off-site? Sure, it's growing in a city, but it's probably looks better than any tree. Well, it looks as good as some of the trees I see in the native range of black walnut in the eastern United States. Is this disease associated with drought? Possibly. Is there climate change involved? Possibly. But don't count on those saving our black walnuts in the eastern United States. I just don't see that as the overriding issue in this disease. So I'd just like to conclude that black walnut is bad news. You need to be on the lookout for this. The only way we're going to stop this is by mitigation and preventing movement of trees. So I guess with that, I'll open it up to questions, and maybe I can start by answering the one by Kevin. Should we advise communities to stop planting black walnut? That's a good question. If I'm in Tennessee right now, I wouldn't be planting black walnut. One hypothesis is that this disease is going to go through and wipe out everything, and since the walnut twig beetle only goes to walnut, when the black walnut dies out, there will be no other host and the population will crash. And then you can go back in with black walnut after the epidemic and will be okay. The thought against that is that we're seeing these 10-inch diameter and smaller trees surviving in Boulder, and we don't know yet whether they're sort of like a typhoid Mary harboring low populations of insects that could then build up again. So I don't know how to answer that. I would be, if it were me, I don't think I would be planting black walnut in municipalities right now. So Debbie asked, can the inner part of the tree be used after the bark phloem and cambium layers are removed? And the answer is yes. This fungus and beetle, as far as we can tell, doesn't degrade or damage the heartwood. And so these trees could be canted or bark otherwise removed and then made into saw logs or veneer or whatever without any problem that we can tell. So I think that's why I say people shouldn't preemptively start felling and selling trees. I just don't think that's a good strategy. Jerry asks, can we survey using freshly cut bolts and then post-season check for beetle and fungi? That's a good question. Dr. Seibold in California did that, and he was not impressed with the results. And I've been at conferences where people ask him that, and he is not very enthusiastic about that. So I'm just hoping that he's going to be able to get that attractant out by next year. I don't think it's going to be ready this year, but I'm hopeful it will be next year. And the question is, do you think this is more widespread in the East? And are we going to be chasing this like we did emerald ash borer? And I hope not, but I suspect that Knoxville is not the only place we're going to see this. Now that people are actually taking this seriously and going out to look for it, I'm fearful that we're going to find other hotspots. Having said that, I think there are management strategies that can be put into place to slow the progression, and if we can slow this down, that gives us some breathing room to develop some better strategies for control. Okay, what if any insecticide, fungicides have been used? And we have a study that we're conducting here in Colorado using imidacloprid, and which is merit, and arena, similar to what's been done on emerald ash borer. There are anecdotal reports from Boulder that merit has slowed the progression of the disease but not completely stopped it. And there are some unconfirmed reports that Safari has been effective. The problem with Safari is it's not labeled, and what we run into with black walnut is it's a nut crop, and with it being an edible crop, that really restricts the types of products that we can use. So things like Safari are not ever going to be legal, I don't think. Triage is not going to be legal, so that's going to be an issue for us. But if I had well, I better not say that. Another question. Yeah, the question is, from initial infestation to visual damage, what is the timeline there? And that's where we have a vigorous debate between Dr. Cranshaw and myself. I think it's about maybe 2 to 5 years. He thinks it may be 5 years, 10 years, or even longer between the initial infestation and the time a tree shows damage. Whatever the case is, it's an extended timeline. And so by the time you see an infested tree with dieback, it means that has probably been established in the location for 3 to 5 years. And that's really bad, because that is going to put us on a track like emerald ash borer, where we're running around chasing the epidemic based on visual symptoms. So until we get this attractant, we're going to be in bad shape, I'm afraid. Did I miss any questions here? Let's see. So there's Bridget asks, I was told at a previous meeting that the beetle and disease are acting differently in Tennessee than in Colorado. I'm basing my observations on a one-day stay in Knoxville, so I could be way off base. From what I saw, the type of damage and dieback was very similar, if not identical, to what we see in Colorado. Now, one issue or one point is that this disease results from continuous invasion by the beetle, and healthy trees with thicker bark are probably more likely to decline much more slowly, or maybe even resist the problem. So the argument is that all of our trees are off-site here in Colorado, and therefore they're always under stress, and I don't deny that. But from what I've seen in Oregon, they also see a decline. Sometimes the decline is more slow than what we see here in Colorado, but I don't know. It'll be interesting to see. But the basic development of it looks similar to me. Quarantines, I don't want to get into that. USDA has decided that this is APHIS has decided that this is a native pest, and they washed their hands of the whole thing. So I don't know what to say. What was the TCD website I mentioned at the beginning? If you go to 1000 Cankers disease, all one word, .info, you can get to our CSU website that has our PowerPoints as well as other information that you're more than willing to use. Yes, it's 1000cankersdisease.info. And feel free to use any of the PowerPoints or any of the images. That's fine with me. Any other quick questions? Okay, I thank you very much for your attention. I hope it's hard to tell with this whether people are paying attention, but thanks.
Thank you very much. It was very informative and interesting. Kind of gave us a sneak peek of what to look for, and really engage people out in other areas, especially in the Midwest, to look for this in the upcoming season.